Okay, hello everyone. For those of you who don't know me, my name is Levi Schrader. I am Mrs. Schrader's son. I graduated from Oklahoma Baptist University with a bachelor's degree in biology, which is really more of my field house, but I'm teaching anatomy at Foundations Academy. So if you've seen me around the McLeod's place or at the barn, um, that's probably why. Um, I'm going to be talking a little bit about photosynthesis in this video, but I'll be making another video covering module 5.2, you could say. So a lot of the cellular respiration processes, they're very difficult to understand even when you get more complex into or get into deeper into things. But I just wanted to kind of help you all understand and help my mom help you all break this down a little bit more. So this is what this video is about. Here's some of the things that I'm going to talk about. We'll talk about what photosynthesis is, just kind of define it. We'll talk about pigmentation. Then we'll talk about some of the parts. Obviously, there's a lot of different components and structures involved in the energy production from light. And then we'll talk a little bit about photosynthesis, photosystems and steps. So photosystems really referring to how light is being absorbed and activated. And then we'll talk a little bit about the Calvin cycle, how we go from just an excited light particle to actual chemical change as far as energy production goes. And then you could say there's a 0.6 to this if we continue on with things like glycolysis and gluconeogenesis, but we're not going to go into that. We'll touch a little bit more on those kinds of things when we talk about general cellular respiration. So for this video, try to keep in mind that we're talking about very plant-specific processes here, and occasionally these rules could apply to some bacteria that have fluorescent characteristics. So what is photosynthesis? Photosynthesis is obviously the production of glucose or other ATP through an excitation of an electron. And that excitation creates a cascade, which will help us produce glucose, so our sugar. This is the net equation. This is commonly what we see when we see photosynthesis. So it's 6 carbon dioxide, 6 water, make 1 glucose and 1 oxygen, which is completely true. But this actually occurs in half steps. So we'll actually see in this process it goes from 3 carbon dioxide to 3 plus 3 water, makes half a glucose molecule and plus 3 oxygen. So this entire process that we're going to talk about today is going to have to occur twice to make 1 glucose molecule, which seems like extra work, but it's actually very efficient to do it this way. So yeah, that's what a net equation it kind of is. It's kind of like saying 1 plus 1 equals 2, when really this more complex equation is more like a biological system, but it still equals 2. It's kind of important for you to know that there's aerobic and anaerobic respiration. Aerobic means with oxygen, anaerobic means without oxygen. You'll see those terms a lot, and hopefully you're already familiar with them. But plants are actually considered aerobic. They need oxygen in order to produce energy, because it doesn't matter about the photosynthesis part of things. It matters about the excretion of byproducts. So it's really hard for them to move oxygen. It's really hard for them to transport carbon dioxide and water into the cell and into the plant if oxygen is not present. So it's not really a process of making energy, it's a process of getting all the components to make energy, which is why plants are considered aerobic. So let's talk a little bit about pigmentation. When you think of pigmentation, I want you to think of coloring. We often see it when we're referring to things like our skin. Someone may have a lighter or darker pigment, especially if they've been tanning or they've been doing other things. But pigmentation really refers to the type of light that's being reflected off of the plant. And that is determined in plants by these things called carotenoids. So carotenoids are color-absorbing molecules or color absorbers. So when we look at a plant is obviously green, which means that these carotenoids are specific to absorbing all the other kinds of light around. So the light that's being absorbed is not the color being shown. It's the light that's being reflected that is being shown. And that's typically only one color. So let's look at this graph right here and we'll go back to it. So in the plants, we have our plant structure, and then within the plant we have these things called chloroplasts, which we'll talk a lot about in this video. And then within the chloroplast we have these things called chlorophyll, which is this weird gel-like structure or substance. And within that chlorophyll, we have 2 different types. We have B and A. Chlorophyll A absorbs a lot of this high energy light. So if we go over here on this graph, this one's going to absorb a lot of red, orange, and yellow light, while chlorophyll B is going to absorb a lot of the darker lights, so purple and blue, these low energy lights, and it's going to reflect the blue. And the reason it does this is it helps, it makes it significantly more stable within the plant while still absorbing adequate amount of light. So the only color of light that's not being absorbed is down here in our 500 to 550, 5 to 600 range, and that's our green light. So that's why plants are green, is because it's reflecting the green light. And then that's what you catch with your eye. So going back here, again, we see that the color that is absorbed is the only color, the only color that's not being absorbed is the color that's being reflected, and then that's giving the plant its color. But for the light that is being absorbed, it then enters these things called photosystems. So based off of what photosystem it enters will determine its function as far as producing energy. So there's two different types of photosystems, photosystem 1 and photosystem 2. Photosystem 2 actually occurs first in this long process, which we'll talk about here in a minute. But the reason it's why it's called 2 is because we discovered it second. We actually discovered 1 first. So don't get them flipped. They're opposites of each other, so 2 happens first, 1 happens second. Keep that in mind. The chloroplast structure is just kind of important to know, especially when we talk about these videos. A lot of the processes that we're going to talk about are going to occur in this thing called the thiocloid, which are these small structures right here. And then we have our stroma or our grana. Our grana are these stacks of thiocloids. So the long ones right here are the thiocloids that connect them. And then we have our chloroplasts, which are the regular rings, and then the grana are the stacks of it all. Electron transport chain, that might be an important term to know, especially for next video, but that pretty much means we're going to be pumping things in and out of areas to help produce ATP, which is our energy currency of the cell. Hopefully you're familiar with that. And then the Calvin cycle is basically saying we're taking energy from ATP and NADPH. NADPH being this this helping hand. It helps move electrons and move hydrogens to certain places. It helps activate and inactivate things, and that's going to help us in the Calvin cycle convert carbon dioxide into sugars. So the carbon dioxide that's in the air, typically it's a bad rap, it's going to help convert that into sugars. So for photosynthesis, it's going to start with this step 1 right here. This is on page 189 of your textbook. So if you need a good explanation, it actually does a pretty good job of it, but here's basically what it's saying in a very simplified form. So in step 1, which occurs here at photosystem 2, remember they're flipped, our water is going to get pretty much shattered into all of its main components. So we have two water right here, and then it's going to get broken up into oxygen. So the oxygen are going to bind together, and then it's going to get broken up into 4 hydrogen and then 4 electrons. And these electrons are going to be sent all the way over here to step 3, which we'll talk about here in a minute. And it's going to pretty much take the NADP and hydrogen and bind them together to activate it and make NADPH for the Calvin cycle. In step 2, so in photosystem 1, it's going to take a lot of these hydrogens and shove them through so ATP can be produced. And when ATP and NADPH are being produced in plants, it's going to activate the Calvin cycle. So, really important for you to know, in order to make the Calvin cycle work and activate it, you have to have 2 things. You have to have ATP and NADPH. Keep that in mind, because we're going to come back to those in the next slide. So in step 3, the NADPH is being produced by photosystem 2, but it's really happening over here on the other side of photosystem 1. So that's being produced and then it's being sent into the Calvin cycle. And then we have this thing called thiocloid diffusion of hydrogen ions. So that pretty much just means that inside the thiocloid, like we see right here, there's a loss of hydrogen over here on this side because we used it to make NADPH. And so what it's going to say is, hey, we need to make it even. We need to make an even number of hydrogens on both sides of the membrane. And so it's going to take the hydrogen and it's going to send it through the structure right here to help produce the ATP. And that's going to be called an ATP pump. and that's going to enter the Calvin cycle as well. So there's actual energy molecule production in the Calvin cycle, but we're going to use that as an investment to make more energy. So with our ATP and NADPH, we're then going to enter the Calvin cycle. And the Calvin cycle is going to take ATP and NADPH and combine it with this molecule called Rubisco. Rubisco is going to be this inactivated form of a carbon that's going to be help used to make sugars, and they're going to undergo carbon fixation. So the Rubisco right here, pretty much on the ends of it, the carbon dioxides are going to be formed in bonds to make little links along it, and it's going to help extend the chain from 3 carbons to 6 carbons by adding 3 carbon dioxides. Keep in mind this process, the oxygens are kicked off, so it's just the carbons being added on. Then through reduction, a lot of hydrogens are going to be shoved onto the chain to help make it longer and undergo a lot of different chemical interactions. And that's going to cost energy, so it's going to cost 6 ATP to help do that reaction. And so when that happens, then we have reduction, which pretty much means that our NADPH is going to be broken apart again, so it can be reused over and over and over. And that's going to give us step 2 down here, our 6-glyceraldehyde 3-phosphate. In your textbook, it's commonly referred to as G3P. And we're going to send 5 of those into the chain to help regenerate this molecule right here, the RUBP, while one of them is going to be shoved off and used for sugar production. So that's why we have to repeat this entire cycle twice, like I talked about in the beginning. It's because we need 2 of them. We need 2 3-GP to produce 1 molecule of glucose. And then that 1 molecule of glucose will undergo a lot of the processes, which we'll see later on, and it'll help produce 30 ATP. So it's an investment of ATP, as we can see here. We invest 9, and we're getting 30 back. So it's a pretty important process. So there's a net gain and not a net loss. And then lastly, we have this step 3, this stage 3, this regeneration of Rubisco. So it pretty much just means our RUBP, which stands for ribulose bisphosphate, the phosphate groups are being kicked off, and then the carbon groups are being added back on. So then we have Rubisco, and it'll undergo the cycle over and over and over. It's really complicated, but the important thing to note is that we're binding things together to make them longer. Then we're adding electrons to it to give it more energy. And then we're breaking it back apart again in a different way so that we can have more molecules to use for more energy production while having some left over to use to make sugar. So that G3P, this one little thing down here, this 1-glyceraldehyde 3-phosphate is then being sent to the mitochondria. There we go. Is then being sent to the mitochondria for gluconeogenesis. For those of you who are familiar with the mitochondria, whether it be in this course or a different one, it's the powerhouse of the cell. That's where all of our energy is being made. So we're now leaving the chloroplast and we're going to the mitochondria. So we're leaving structures to make more and more energy in a process called gluconeogenesis. That sounds like a really big and fancy word, but we'll break it down here. Gluco meaning glucose or sugar, neo meaning new, and genesis meaning to create or produce. So all that word means is to make new sugar. So gluconeogenesis, to make new sugar. And that's when we get to the final part of this video, which means we're building up to breakdown. So everything we've talked about is creating these larger structures to then be broken down and release more energy to help us create more energy. Which sounds really confusing, but it's the best way as organisms one can get a return on its initial investment. So it seems complicated, it could take a lot of shortcuts, but it's not about doing things fast, it's about doing things at the highest level of efficiency, which is what plants are really good at and why they undergo photosynthesis. So hopefully you enjoy this video. I will post another one, or I'll make another one over the processes that continue on within the mitochondria, but that will be more generalized for not just plants, but for all different types of organisms. So thank you.